Howdy everyone, you're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a Ph.D. program while trying to stay sane. We also often invite faculty from our interdisciplinary program here at Texas A&M and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M. In this episode, we'll talk to Dr. Qing Sun. Dr. Sun is an assistant professor in the Artie McFerrin Department of Chemical Engineering at Texas A&M University. She received her Ph.D. in chemical engineering from the University of Delaware and did her postdoctoral training at the Synthetic Biology Center at MIT. Her lab uses synthetic biology to improve human health and the environment. My name is Sarah, and I'll be your host today.
My name is Samantha, and I'll be your co-host.
And I'm Qing Sun. Yeah, thank you for inviting me.
Thank you so much for coming on. I would like to start by asking, what does a typical day look like in your lab?
Oh yeah, so typically day I would cover yesterday and today. For example, yesterday I went teaching. I teach from 11 to 12:30, and then I host office hour after that. So before that, I was mainly preparing for my class. After that, I have several meetings with my students talking about your progress if you need any help. This morning, I came in, had collaborating meetings through Zoom with my collaborators. And afterward, we have group meeting. Today is, because I'm traveling on Friday, we have group meeting. I had several sub-meetings with my students afterward, and here I am. So basically, research and teaching is what I do most. And after I go home, I have 3 cats at home. I just play with them. Yeah, that's my typical day. I think I'm going to go swimming right after this. So I want to get some exercise. I don't want to be sitting down all day and doing nothing. Yeah, I want to improving the world to be a better, healthier, and more environmental friendly. I also want to be healthier from my side.
Yeah, sometimes it can be hard to take care of yourself when you're so focused on your research.
Yeah, especially I sit down a lot of time for proposal writings, so I want to exercise a little bit more. Yeah.
Why did you choose to come to Texas A&M University to do your research?
That's a good question. So I think A&M is a really interdisciplinary university in terms of research areas and in terms of collaborators I can find. In terms of students, like this genetics program, that they have diverse interests. So I started, it's actually quite interesting. I started my undergraduate as an environmental engineer, like my bachelor degree. I did my master in environmental engineering, like PhD in chemical engineering, as you introduced. And then did my postdoc in bioengineering, synthetic biology. So after those, I feel like I really need to find a place where I can do environmental research, health-related research, based on biological background. So I found the chemical engineering at A&M is a great location because it has diverse research areas, open-minded. The students in there are interested in different research areas. So after looking at those, I feel this is a place to do research, and that's really my passion. I like this little town. It's not a little town, but it's a town college station. That's why I'm here. Yeah.
Did you face any challenges when you were becoming a professor?
Yeah, of course. I think the challenge mainly is how to establish myself, because I have a diverse background, so it's hard to be focused, how to say what I do. So I think in the first 2 years, I only have 2 or 3 students, with limited power, research capability. What can we do? So I think at that time I was like, what am I? What can I do, right? So I did talk to multiple collaborators. I did talk to my mentors. I do have mentors, and also my students, and also based on students' interests. I think the first 2 years was a critical time for me to find what my true interests are in terms of specific project, and how do I want to be seen in this area. So I think that's the 2 things. That's the thing that I struggled a little bit, but I think I am more and more clearer and clearer about what I want to do in the future. Yeah.
So why did you get involved in synthetic biology? Why is that something that you decided to pursue? Mm-hmm. And I guess then as a segue, if you could talk a little bit about some of your research going on in the lab.
Oh, yeah, of course. Again, I think it started with my environmental engineering background. So when I was a high school student, when I first got into college, I was really passionate about like how can we save the earth, because I saw a lot of pollution when I grew up. The river beside my home just got like super dark. It's become like not clear anymore. I cannot even go swimming in there anymore. I used to go swimming. So I care about the earth a lot. So when I got into my PhD, I started looking into what kind of things like can help me rebuild the sustainability of the world. So I got into a research lab, Dr. Wilfred Chen's lab at Delaware, where he has been doing a lot of biofuel like research, where you can convert cellulose to useful biofuels. So that was really my passion in there. And also I feel chemical engineering also solve a lot of problem by improving the efficiency to reduce the waste releasement. So that was super helpful. And throughout my PhD, I found that there are many other things that I can do with that toolbox. So basically, like by the end of the PhD, I switched my research project from biofuel production to actually cancer diagnosis. It sounds like two totally different research area, right? But the fact is I can share the same toolbox. So I was super excited when I found out even using those cute toolbox of biotechnology, synthetic biology, I can do so many different things. So I started merging them together. So when I was like applying for my postdoc, actually I started looking at what is really my passion, and synthetic biology really is a toolbox I want to use for my future career. So when I was applying, I was focusing on doing synthetic biology and got into Dr. Tim Lu's lab at MIT. Again, he's a lab with diverse research interests, including environmental, including pharmaceutical applications. So I found that this is really for me, because I always know I want to do biology, but with this biology, what I can apply it to is what I have been trying to figure out, and those two labs really helped me. So from there, after I started my lab, actually I'm like emerging, like putting these two together. So our lab really have two big areas. One is bioenergy and sustainability. The other is biomedical. So the first area, for example, like I'm super excited about plastic degradation. So I always post this by asking questions. So do you know we have plastics everywhere, even in the forest, oceans, even in some of the animal blood. So that is a serious problem. So with that, so I'm trying to use synthetic biology toolbox to solve some of the plastic like degradation and upcycling problems. So the second question I always ask people is also like, do you know coral reefs? We're losing coral reefs. So only was like I get into this area, I know like we have lost like 50% of the coral reefs globally since 1950. It's really fast speed. And if we don't do anything, we may lose 90% of them. And in 2050, it's not that far away. And also coral reefs is very important. It's not only because it's beautiful, like where we can go snorkeling for, it's also because it's a home for 25% of marine animals. If 25% of the marine animals lose their home, what will happen? Right, so I'm so concerned about this. That's why it's another bioenvironmental energy project I have been doing. So the third question, I always ask question, is that we have many mRNA vaccines going around, especially during pandemics. So it works very well, but the question is, do you know that it's actually hard to store them and distribute them? We need -20 and -80 degrees centigrade storage conditions, so which is not a super, I would say, sustainable solution for biomedical applications. So like what we're doing in the lab is we have an algorithm, like deep learning approach together with experiments, trying to improve the thermostability of mRNA vaccines. So I think I asked 3 questions about plastic degradation, about coral reefs, as well as mRNA vaccines. So I think that also reflects that my background of using synthetic biology toolbox to actually apply them to different areas. So that's where I am now. So I'm quite excited about what we can do using our toolbox. Yeah, hope that addressed your question. Yeah.
So as you got into it, you have a very diverse set of research projects.
Yeah.
So what kind of challenges do you kind of run into as far as overseeing all of those projects, staying up to date on all of the science that might be going on in those fields, and how do you kind of overcome some of those challenges?
Yeah, I think the main challenge is, again, even if we have the same toolbox, right? Same toolbox using DNA, RNA, and protein, Like for example, the mRNA vaccine is RNA-based project. The plasmid degradation is protein-based. Coral reefs project is bacteria-based project. So we all have the same toolbox, but still every time you will run into a new platform, that new model animal or new model organism you need to use. So sometimes we're like, I know this how this cloning works, but I don't know how this cloning works in a new organism. Sometimes it's even non-model organisms. So that was a challenging one. I have to convince my students, hey, it's super exciting and it's feasible and we can do it. So those challenges are mainly to apply these things to new organism. So what I usually do to solve it is to find a collaborator or free consultant in academia because academic people are so nice and so collaborative. So like when we were working with C. elegans, I reached out to Dr. Garcia. So he's like, can I meet you? Now he's like, of course. And I went to talk to him together with my postdoc. I was like, I want to work on this, but we're not so familiar with those C. elegans things. So how can work on it? So he actually gave some really quick tips, and we actually sent our student to his lab, learn how to culture C. elegans. Like the coral reef project, I have no idea about how we can culture enemies in the lab. So we actually have a collaborator from UC Riverside, Dr. Qing Sun, where we talked to him, and he sent us a lot of enemies as well as algae. So that's how we solve most of the challenges we run into when we want to apply our toolbox into a new platform. So it has been working, but I imagine if we are interested in more projects, we may need to find more collaborators. But that's the main challenge we run into, yeah.
Yeah. So do you find then as well that your lab has more of a collaborative environment within itself as well, because they each kind of have their own specialty, they can kind of help each other out?
Yeah, exactly. That's what we found. So basically, we have several subgroups. You can tell we have the plastic subgroups, we have the coral reef subgroups, we have the vaccine. We actually have another living vaccine that is growing subgroups. So we usually have subgroup meetings. So each subgroup meeting, we mainly have 3 or 4 students. They really collaborate. They help each other. But meanwhile, we have group meeting where, like, if you talk about your challenges, the people with other expertise will come help you. So basically, they work on their own projects. I would say that everyone has their own leading projects. It's not like you work on like several projects, like all your leading project. You may have 2 of your leading project, but meanwhile, if your two sets can be used in other project, I can see my students are really happy to help each other. We actually build a super friendly environment. Every time I have rotation students or new students, I will have a really nice environment. I want to make sure I don't destroy it. Yeah. So basically, it's all very collaborative because it's all similar toolbox used in different organisms, but the base like I would say mechanism are quite similar. So they always help each other to troubleshoot.
Yeah. I mean, I rotated in your
Yeah,
lab,
yeah.
so I know that your lab is very friendly and they do have a very good environment, yes.
Yeah. Thank you. Yeah. I make sure everyone got into the lab at least have a nice personality, like a fit into the environment. Yeah.
Right.
Mm-hmm.
Yeah.
Do you think the collaboration helps with managing your graduate students and your undergrads? 'Cause I believe you have a decent amount of people in your lab, right?
Yeah, now I have 10 people, like 8 graduate students and 2. It's not a super big lab, but for me, I think having this collaborative environment is super helpful. For example, if the first student, like the junior student came into the lab, sometimes they came in knowing a lot of biology. Sometimes a chemical engineering student, you know that, they basically know nothing about biology. So in that case, they usually need a lot of training and like those kind of mentoring. So so that's one way. I also train the leadership of the students in the lab. I also senior students to train the junior students with those basic techniques, because the way like the senior student, when they talk to the junior student, they have to say and talk about how this assay works, how this protocol works. Sometimes they found, I don't know about that, so that they need to go back and think about it. They also teach about logics, about how to design experiments, negative control, positive control, and during the teaching, the senior students also learn, like maybe find something that they did not realize before. And then after those training, the junior student will be on their own. They have to like figure out the logics behind their own project. They also need to do presentations. So they present to the group. So the group will help them like figure out, is it really the big problem you're solving, or like how can we like ask many questions so that you can like figure out what's really going on, or like at least do well in your committee meetings. So I think those collaboration, like a collaborative environment, is super helpful not only for junior students. Like we always think junior students are the one that benefits from the system, but I think the senior students also learn how to leadership, how to mentor students, and meanwhile fix what they thought they know but they don't know. So I think this environment is helpful for both like junior and senior people in the lab. Yeah, that's what I found.
Has anyone in your lab found anything particularly surprising or exciting recently?
You mean any personnel in the lab?
Yeah, any personnel, you or any personnel in your lab, has there been a surprising or exciting discovery that you've come across with your multiple lines of research?
I see. That's actually a really good question. So I asked 3 questions with plastics, mRNA, and coral reefs. Actually, the mRNA project is actually from the lab. Oh. It's not from me. So it's like I got a student, he came in, and he was talking to my other people. I was like, you come in, we have a project for you. And then they started talking. So 2 students started talking. They were like, DNA is super exciting. Maybe we can use deep learning to work on this together. So they actually 3 students, they were talking together. And then they were like, we can use deep learning to predict the DNA functions. So they were very excited, and they come talk to me. I was like, oh yeah, that's actually something we experimentally very strong, we can validate. But meanwhile, if you have a deep learning tool can use, please go ahead and develop this algorithm. So they started working on this together, and during the process, pandemics happened. And mRNA vaccine started showing up, and we have that algorithm ready that is actually predicting nucleic acid functions. So we found using those, we found that there's a great application for mRNA thermostability prediction. And we found a data set for the model training, and then we move forward with that. So that's growing into a really big directions in the lab. So every time I talk about this one very proudly, but it's actually from discussion within the lab members, and it's actually from collaboration within the lab members, because we have experiment people talking to the algorithm person. And also one thing, the deep learning people came in with 0 biological background. He does not know how biology works, but we taught him. He asked all the questions, but meanwhile our group has no knowledge like me. We don't know how deep learning works, but he actually showed us how this works. Now I know the basic algorithm that we can use. We know how to train the data, how to clean the data set, how to balance it up, and how do we predict new things. So I think that really in this is a special case of showing how we can use the surprising, friendly environment to create new directions for the lab. So I'm quite encouraged of that. I would say you have your project you're leading, but always feel free to bring up new project to me, because we're very diverse. I send you to conference, feel free to learn new things, and we discuss. Of course, I have to sit there, help them. What's the motivation? What's the feasibility? What's the novelty we do here? But overall, I think the students will learn how to develop new project after they graduate. That's what I hope for. Yeah.
So do you find there's good correlation between your algorithm and experimental results as far as mRNA stability and all those?
Yeah, I would say, so we have the algorithm, we predict mRNA with better stabilities. So we found one tool that works very well, and that actually beats all the other algorithm out there. And there was one protein we predict is not as good as others, but overall, if we predict it's going to be more thermostable, experiment-wise, it's more stable. It's just sometimes it's much better than other people's algorithms. Sometimes it's a little bit similar. So that's what we found. But overall, we can predict them very reliably. And nowadays, we can make them super stable, but maybe something you don't want, we make them super unstable. So to extreme end. So that is actually another thing we're doing with bacteria. We want them to tune their expression levels in bacteria, so we can predictably make them more stable and higher expression, and no stability at all and low expression. So it's quite predictable so far, yeah.
So are you working with any type of specific sequence or specific protein, or are you just kind of getting a broad...
Yeah, the first thing is we always like to start with green fluorescence protein. So green fluorescence protein, most of biology people know it's a cute tool you can test, and they have fluorescence. It's easy to measure. So currently what we're doing now is spike protein. and not part of spike protein. It's called RBD. So that can also have the immune triggering capabilities. So RBD looks like it's working, and then we're going to expand them to longer sequences like spike proteins, like 2 kilodalton. And then we're going to, other than spike protein, which is the COVID vaccine, right? But COVID is almost, hopefully it's gone forever. I know it's still around. But next we'll also look at mRNA with different functions like CRISPR, because we believe by improving the stability of CRISPR, we can improve the gene editing efficiency. So that's something we're also working on. And also we're looking at different RNA with different functionalities. But those are several examples. Like we believe it's not limited functions, but like we can actually have a strong platform that can be applied not only to those pharmaceutical applications like spike protein, like CRISPR, but also we can apply that to bacterial engineering. So that's many things that we're working on. We have those 2 manuscripts we're ready to submit. Yeah.
That's awesome. That's exciting.
Yeah. That a lot of things happen after your rotation, though.
Yeah.
I know, yeah. Always check with me what's going on.
Yes.
Yeah.
Lots of things can happen in a year.
Yeah, I know. Yes.
So you had mentioned you'll be having some publications coming out. From the side of being a principal investigator. Is publication something you think is important for your graduate students to get while you're in school?
Yeah, so that's an interesting question and exciting question, actually, that I have been thinking about. For example, when I was a graduate student, I care nothing else but my research. And the paper is the outcome of my research, right? So I can go to conference, talk about it, and I can do super high impact research that excites me, but I also want people to see it. So like conference, presentations, and papers was all I care about. After I get into academia, there is something else you need to worry about. You need to feed the group. You need to make sure there's enough funding to support a group. So I started thinking, how can I get more funding into the group? And also, like, going to conference will spread what we're doing that will help. So I'm trying to find a balance between the funding and between the presentation and the conferences, because like for students, most of people they have no idea about funding. They're like, you got me in, you just feed me until I graduate. Right? I just do whatever I'm interested in or what you tell me to do and get a paper out. So, but from my side, I need to think about how to get proper funding for the project we're super excited about. But meanwhile, I also care about papers and presentations because I know that's what students care about. I know that's the outcome of the funding, I would say that. So I'm trying to find a balance out of it. I believe, so I would say I care about papers because that actually related to what my students are doing and related to what's the outcome we have. But again, that the research itself is the most exciting part. So I want to do high impact research. For example, I want to save the coral reefs if possible. I want to improve the mRNA stability. We also want to do some living vaccines. But the idea is how can we help more people, help the society, help the earth, save the earth. So that's the most important part, and the outcome might be funding papers. So I always tell my students, like, you should be excited about what you do. And if you have the right hypothesis, if you have the right approach, you should be able to produce a paper, at least, right? I know we have paper requirement for graduation as well. That's part of, if you're well-trained, you should be able to produce those. But the core should be the research, yes. Hope that answers your question.
Oh, it does.
Yeah.
Absolutely. So do you have, or do you see your students lean more towards academia or industry, or is it a pretty even split?
Yeah, so most of my students are from chemical engineering, so I can see they come in with the industry match. I do have 1 or 2 students interested in academia, so I would say 2 out of 8 or 9. So I see, like, I'm not trying to change them because I feel both areas are super exciting. So like most of my group members are interested in industry. Some are interested in biotechnology, in general any biotechnology. Some of them super interested in like pharmaceutical areas. So my first student is quite interesting. My first student actually went to Merck and as a biopharmaceutics. And second student actually got a job at Wall Street.
Oh.
Yeah, that's quite diverse, right? My
Yeah.
third student, he's interested in academia. He's still in the lab, so I don't know how well that will go, but maybe at least a postdoc. So I respect everyone's individual interest. Like you come in with the interest. I also respect your interest may change during your PhD. Maybe you came in, you're like, I have no idea about academia. I want to do industry. But research is so fascinating, right? I want to do academia afterward. Or some people came in with academia, but then he saw me struggle or something. I hope I did not show it. But then they were like, okay, I don't want to do academia because of you. It's fine. I did not do a good model. But I try to talk to them, like to see if your interests have changed and how can I help you to get your dreams. Because I feel like a lot of students come in like, I'm going to start for my PhD and then get a degree and do whatever. But my point is, after you get your degree, you should really think about how can you achieve success in both your personal life and career dreams. So I cannot do much about your personal life, but your career, at least we can help you with the right project and maybe the potential like internships to get you into the what you want to do, if that's what you think you would like doing. So far most of them are interested in industry. So like some area are easier to get a job. So I would say they know what they are doing. And if they want to get an internship or co-op, so 1-2 times, we can always talk about it. So I think I'm trying to help them to find their right career. So whatever is best for them, yeah.
So what made you want to join academia versus industry? What kind of fueled that choice?
Oh, yeah, that's a good question as well. I always tell the story about how I was not determined until I finished my postdoc. I was, as mentioned, I was an environmental engineer when I started. I was like, I want to save the earth. I don't care about what I want to do in the future. I just want to do some research now, undergraduate and a master's. And after getting into PhD, I was like, how can I not save the earth, but trying to reduce the waste production as a chemical engineer, and how can I use biological solution to save things? And I was not thinking about what I want to do. And when I was close to my graduation, I was like, oh, everyone got a job, so what do I do? And I think about it, I talk to several industry people. I feel like they're doing exciting things, like my friend doing vaccine production, my friend like doing biofuel production, using pharmaceutical production, using reactors. I do feel like it might not fit me very well because I want to do more fundamental research, like upstream applications. So I was like, okay, let me not get a job first, industry job. Let me go to my postdoc. And that will give me some buffering time before I have to make a real decision for my life. So I went to postdoc. And during postdoc, I started realizing that I talked to more people because especially after postdoc, I started thinking more about my career. I've realized there are so many options. You can do a patent lawyer, like as an engineer. You can do a consultant, and you can start a startup or join a startup, or you can go to these big companies, or you can be a professor. So I actually started talking to more people about what they want to do. And one thing I found that is not for me, again, is industry. That is not like saying it's bad because it fits many people and my colleagues are also happy there. I just find it's not for me. I want to do those fundamental different things that I want to do. So it took me a while to figure out until the second year of my postdoc. I was like, okay, I'm sure I want to do a faculty job. So academia is what for me. It took me such a long time to figure out. I could not believe it in the beginning. I thought I could have known what I want to do and being super successful at my year 30 already. But turns out I only figured out like I want to be an academic person at year 30. So I started applying. I got into, and I exposed my age accidentally. But anyway, that I decided to go to academia, and I got this super exciting opportunity at A&M, which you asked me earlier. Why do I think A&M is a great opportunity? I think it's this place where I can do all my research I want. It's so interdisciplinary, and I got students with interdisciplinary interests and background. So that's where I started. So it all come together like thing. So I always tell my students, don't worry about not being able to find what you want to do now. We always have this career conversation, like what do you want to do now? And does it change from last year? So maybe you change it again next year. So don't worry about this because your career dream may also change throughout the throughout your life, right? In the future, you can always correct it, but to find what is best for you for now, and for after you graduate is very important. So it took me well to figure out, I would say, and I encourage my students to not be in panic if they don't know what they want to do. They will figure out. Yeah. So that's my experience. Hope that helpful.
So what was your experience like transitioning from being a postdoc into a PI?
Yeah.
I'm sure that's a difficult transition, but what was that experience like for you?
That's a good question. So I think the first thing from postdoc to PI again was the funding thing. Like when I was a postdoc, actually I was a student leader of the doctoral project. I need to organize all the updates and send the summary report every 1 month or 3 months stuff. And even that time, I did not realize what PI life was like. So after I started the first thing you need to feed the lab, right? So you need to write a lot of fundings, proposals to make sure that you have enough grant to support the group. Second thing is you started from taking care of your own project, right? Even at that time, we have many collaborations. I need to ask them what's your update stuff, but taking care of your own project to start mentoring several projects. So that is another big different thing to me. So think about one project straight line. So you can always I want to check what you're doing, but you're not doing it, right? So you just out of interest. But now I need to make sure I know all the details, everything. So that's the second thing that is different. The third thing is definitely teaching. I used to be TA multiple times. And once we started our PI life, I was thrown into a room and you can start teaching. And that was challenging for me at that time because I know I got all the notes from other people, but talking for 1 hour continuously and think about what's next and the equation should be right or something. So that's another challenging. I would say, yeah. So that's what I said. So I think teaching, funding raising, as well as mentoring different people is the thing that is challenging. And also, I'm also learning now, so it's not I'm settling. I'm still assistant professor. I've been here for 6 years. One thing I noticed is that every student is so different. When I was a student or postdoc, every personnel in the lab is so friendly. I like them all. But once you get to the PI level, you find every student is different. Some of them need hands-on mentoring. Some students are really hands-off. They want you to be staying away from them, so they can do their thing, but they can figure it out. And some students are more they want to do some new things. Some students were you tell me something and I will do it. And some students are communicates everything. Some student does not like to communicate. There's so many things that is in the lab that I'm also learning how to have a happy lab, as mentioned, friendly lab, but meanwhile being productive lab. So I'm still learning those. So as mentioned, I think from postdoc to PI, I think funding raising, mentoring, and teaching was several things that I struggled. Basically, if you're a PI, you have to do everything. You need to do your financing, recruiting, and management, everything. Yeah, but those things are kind of special for me. That I struggled a bit. Yeah. Mm-hmm. Hope that answered your question.
Yeah, absolutely.
Yeah.
So, I understand that being a PI is a lot, and so do you have any hobbies or things that you like to do to kind of help give yourself a break any of those kinds of things?
Oh, yeah. Actually, that reminds me, I only talk about bad things about being a PI, which is not good. I do enjoy doing research, and I mentioned about how I got here, and I enjoy doing different research, environmental and biomedical. But I do realize that after research, I need to have my own life. So I do have 3 cats. Name is Kit Kat, Bullet. I forgot the other one's name. So three little girls. They are very cute. They wait for me to go home and stay on my lap, so I can pet them, feed them every day. I do like traveling, so that's why I mentioned in the beginning, when there is conference, I like to go explore the area, Orlando, New Orleans, San Diego, everywhere. Actually I want to go globally if there's a chance. So I like those. And I do go to gym a lot. So I used to go a lot more. I go swimming, badminton, I have my personal trainer who like yelled at me to work harder and harder. So I'm also trying to learn new things like tennis, those kind of things. My husband do the rock climbing, so I'm trying to learn a little bit, and I know I don't like it, so I gave up already. So there are several things that I think I'm trying to get in there and enjoy, because I believe like being a PI is like being a student. It's a marathon. It's not like you run really fast, you finish. I think it's a lifetime thing. So I want to have a balance between the work and life. So like I want to make sure I stay healthy and work longer for everything I talk about the lab. Yeah.
So how do you encourage your students to have that work-life balance as well?
It's hard. That's the thing. Like it's hard. I always tell my students the most important thing is work smart, not work hard. So you think you will stay overnight, you will get new results. But the fact is that if you stay overnight, you second day or maybe for the next week you cannot think. So I always tell my students how you can work smart and not work like super hard. But work hard is part of PhD, right? So meanwhile, I don't want to have too much interference of their life. I don't want to be like go find a hobby. That's something like they want to figure out on their own. I believe if the students have a good planning, scheduling for their experiment, they will able to find something on their own. So like during our lunch, like group lunch, when we talk about research and exchanging new ideas, I'll talk about many other things. I do ask, what's your hobby? So I do believe everyone in the lab have a hobby. Even sometimes they talk about, I like to clean the house. That's a hobby, right? So I was like, oh yeah, I don't have that hobby, but I respect that. So they don't have hobby like some people like playing piano, those kind of things. So I usually don't encourage them to like, if you don't have hobby, I encourage them to find a hobby. Just like, I believe they can figure out how to do the work-life balance. One thing we don't do in the lab is you have to be here at like 9 a.m. to 9 p.m. We never do that. I believe the student will figure out their theme and what they need to do before, like for the work and for the weekend, like whatever they want to do. So I believe the students occasionally come talk to me that, oh yeah, they want a break like for 2 or 3 days. So I'm always okay with it because I know they will, I know they are working really hard. That's the thing I have belief in my lab. So like, I believe they have, hopefully they have work-life balance. I never want to go too personally with them, but I believe they can figure this out. Yeah, but again, it's from me. Hopefully they don't think we have too much pressure. I don't believe so, yeah. So I think they have a work-life balance. And I always tell them that I go to gym a lot. So I believe they also figure those things out, yeah. But I don't want to, hey, can you go to a gym with me? I never do that. Yeah, it's hard, especially if some people don't want to exercise, yeah.
Do you think your graduate school experience helped shape how you treat your graduate students?
Yeah, I think it's actually both my graduate life and the postdoc life, because my graduate advisor, he's responsible, and we have 3 meetings per week.
Oh, goodness.
Like I see him a lot of, sometimes like once per day at least. So I think it's a good thing for me because I was a graduate student. I don't know what I'm doing. I want to see him and talk to him more about like, is this experiment okay? And if I need to make changes, or talk about new ideas, talk about new papers that is just out of in this area. So I think that's helpful for me. And when I got to my postdoc, it's another extreme. My postdoc advisor, like when I first joined there, they have like weekly meetings and then become monthly meetings, and then he become more busy there. So I saw him every 2 or 3 months. But I was a postdoc. I believe a PhD student saw him more. So that actually is good for me as well because I was a postdoc. I was independent enough to know what I need to do for my research, what I need to know for my work-life balance. I was more independent at that time. So combining those two together, so actually shaped my way of lab mentoring. So I was like, as I mentioned, some students like more hands-off, like mentoring, some like hands-on, like mentoring. So I'm trying to sort it like train the student differently. If you want to meet me more, I can schedule more meetings with you. If you want to meet me less, like other than the group meeting and subgroup meeting, I don't meet you at all. So like you can figure it out. So those really help me with my mentoring styles. I think it's super helpful. Like I know how it is like working in those more, I would say, hands-on lab and hands-off lab. And meanwhile, like because I did my research area shift during my PhD from the environmental to biomedical, so I understand my students, some of them may have diverse interests as well, and that's how I shaped my lab. So like if you're more interested in environmental, just talk to me and we can find a fit for you. And if you're interested in biomedical, you surprisingly you can see different people from different programs have those similar interests. So if you're interested in biomedical, if we have an opening, that's a great position for you. So I'm trying to find a balance between those. So like, and also if the student in between, they want to do something different, as long as you can finish your main project, I'm very happy if you want to try something new. But again, I need to like sit down with you because sometimes you're less experienced in determining the motivation, feasibility of the project, and I can help. So I would say for my PhD and postdoc experience, both help me with my mentoring style and research area shaping. I would say that helps a lot and benefits so much from it, from those experiences. Yeah.
What would be your biggest recommendation or like, yeah, I guess recommendation for current graduate students, just, I mean, whether that's just work-life balance or in their research or like what kind of advice, that's the word I was looking for, advice.
Would you
have?
Yeah, that was a thing I was thinking about throughout my PhD. Yeah, like how can I have my PhD life like more fruitful, less stressful? So I have one advice. I don't know if that applies to all, but to stay positive. I mean, especially it's really hard because when I started learning first doing how to do cloning, I feel for the whole 1 month, nothing positive came out. I was like, when I talked to my colleague, I almost cried. I was like, it's not working. And that colleague was, are you crying because your construct did not work? I was like, because that's my life, right? So like you do nothing but research in the lab. And then after that, I figured out feeding a construct is so common. Like you just need to troubleshoot more. I mean, eventually it worked, but I would say like if I talk to myself at that time, I'd be like stay positive, stay focused on the problem, rather than like seeing emotional fluctuation. It's really hard to keep care of that, but stay positive. Think about how you can make things work, or keep things right, rather than emotion fluctuation. And I think one thing really helped me is work-life balance. Like I actually like to travel if I have like those holidays, and I started exercising a lot during my PhD. Even I did not do that much during my undergraduate. So have a work-life balance will take your mind away from your research a little bit. Like when I was playing badminton or swimming, basically I cannot think about my research. I was like, I cannot let that ball drop. I have to pick it up, or I cannot draw. I have to keep swimming. So those things actually help me my mind clearer. So that actually helped me keep positive. So if there's any advice, I would say like stay positive. Think about it as a marathon. It's not like 1,000 meters you have to finish running in 2 minutes or 3 minutes. So it's something you have to think about. I need to have things right. If something bad happens, like experiment not working, if you have a toxic colleague or something, stay positive. Think about what you can do to make it right, rather than like being really occupied by all the bad feelings, because that will destroy your PhD. PhD is not easy, but definitely it's doable, right? Think about what's going on with your colleagues, everyone. They actually were able to do it, so you can do it. But don't do it in a super sad way. Like you're like crying, winning not in a crying, think about everything is wrong, so this kind of, it's not good for you. I think stay positive and trying to find something like you can do on the side. I know a friend, he's like fixing watch every night. So like he goes home, clear his desk and do those. And I have another friend who just go home and clean his bedroom. I think that's another hobby he has. And then he turn off the light only with the lamp on, and then he read his books with a wine. Only advisable if you're at that age, beyond that age. So he has some wine open, reading some books at night. So that also helps. And also go out with your friends, enjoy those environments, right? So especially like your friends is all doing research, and if talking to your PhD friends and the master friends, so like they have a lot of things to share. So like talk to people. So don't be on your own and think about your research not working. If the research, if every experiment worked, you should have finish your PhD in 1 year, right? And it's not happening. So most of the time it's going to not work, and you'll troubleshoot. That's what you learn. So stay positive, find a work-life balance. I think that really helps with your PhD. And also your interests could change, right? So like me, environmental to biomedical, and if you're meant from industry to academia, or academia to industry, it's all quite normal. Just stay positive and figure your way out. So that's my suggestion.
Yeah, I think that's good because at least for me, and I think this is a bit of a universal experience, but I go home and the research is still in my head.
Yeah.
And it's hard to not think about it, and you lay down at night and that's what you're thinking about as you're falling asleep. So I think it's definitely good advice to try and find something that kind of occupies your brain space.
A little bit,
so that you're not constantly dwelling on it. Yes.
I mean, if it's working well, just let it go. Just think about it all night. Think like it's working, it's going to be great. I'm going to get a high impact research done. I'm going to have a nice paper. I'm going to graduate in 3 years. Let it go. But if something not going well, then you're like, okay, let other things come in a little bit. Like think about how to troubleshoot in a more calm down mode rather than like panicking. It's not helping. I would say focus on the problem itself. Yeah. Mm-hmm. Yeah, hopefully this is helpful. I did not do very well, to be honest. I was like, no, it's not going to work. No, I'm not going to graduate. So it happened to me multiple times. That's why when I got to this stage, I'll be like, you're good, you're fine, you will do well. So, but don't let yourself think about it too much. Just like focus on how to troubleshoot, how to make it work. Yeah. Mm-hmm.
I resonate really hard with, switching. My undergrad research, I actually studied viruses in coral.
Okay.
So,
really?
So I'm very interested in your coral
yeah.
research, and now I'm over in psychiatry, so very big difference. But
okay, I see.
I understand what you mean, and I can definitely piggyback on that. It's sometimes where you start out is not where you're really meant to be.
Yeah, especially like even after your PhD, you can still change your research area, right? So I think the PhD training is really about your logics and your like figure out new problems and your approach to how to solve new problems, how to make an impact. You can use the same thing to many other new things. That's why a lot of PI, if you look at them, they started working on something as their assistant professor, but then they shift to a totally different research area. That's all possible, but it all happens, and according to your interests and your experience, it all happens. But again, stay positive is quite important, and keep your passion. Don't get killed for that. Like don't with all these energies. I think positive energy is very important throughout. Yeah.
Yeah, and I will definitely say, I mean, I've met a lot of professors here, and every single time I see you, you are the friendliest, happiest person in the room. So I was really excited to hear that we were going to get to interview you today.
Oh, thank you so much. I had a great time. Actually, you asked really good question to refresh my memory about my PhD and my postdoc, including research, including like mentoring, including all my like crying moments, my construct did not work. It all come back to me. I think it's a great experience sitting here and talk. We should talk more.
Yeah, absolutely.
Yeah? Okay.
Yes.
All right, guys. Today's episode was hosted by Samantha Foster and Sarah Ingram and produced by Anthony Matarazzo. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, YouTube, and X at Genetics Podcast. Thank you for listening, and we hope you'll tune in next time.
Howdy, everyone. I'm one of your Blue Jeans and Boots hosts, Anthony Matarazzo. Here to let you know that the interdisciplinary graduate program in genetics and genomics at Texas A&M University is currently accepting applications for our PhD program. Whether you've listened to our podcast for a while or are new here and have been thinking about going to graduate school, check out our flyer posted on our website at genetics.tamu.edu. Our program is truly interdisciplinary and includes developmental biology, evolutionary genetics, bioinformatics, plant genetics, medical and applied sciences, and so much more. Come learn from researchers in over a dozen departments and find your scientific passion today. The application is free and the stipend is competitive. Learn more on our website at genetics.tamu.edu.